What a delight to be here today, to be able to talk to you all. I first came up with this talk by request from the staff, and I really have been thinking about the staff so much over the past year. They've done so much for us, and they've worked so hard, and so it really did turn out to be a labor of love, and I'm thrilled to be able to continue to share it to you. So this is going to be great. I'll tell you a little bit about my background, which is that in addition to that cooking class, which I love so much, I also teach, or have taught for many years, a class on applying engineering concepts to medicine and physiology. And in my own lab, I study the immune system, in particular the innate immune system, and the interplay between hosts and pathogens. And so it's from that context that I'll be talking about vaccines today. Briefly, when we think about what our immune system's job is to do, right? It's basically three things, right? It's supposed to recognize, destroy, and remember the different invaders, right? The different infectious agents that might enter your body. And all a vaccine is supposed to do is just supposed to help improve and accelerate that process. Vaccines actually have a now 300-year history. So if we go back 300 years, to the smallpox epidemic in Boston, you'll know, right, that this was a horrible plague. It was impacting very many people. And so what happened, there's this kind of amazing event that occurred. And I think a lot of people might imagine that vaccination, or in this case, inoculation, came from Europe or, from America, but actually it came from Africa. So the original idea of inoculation, here's the story told by Cotton Mather, who was a leader in Boston at the time. Many months before I met with any intimations of treating the smallpox with the method of inoculation, I had from a servant, and I have to interject here, this was actually a slave of his, he heard this account that it was being practiced in Africa. And so Wanzamis, this slave, said that when he was asked if he had ever had the smallpox, he said, well, yes and no, and then said that he had had this operation in which he had received the smallpox from someone else, right, in such a way that he would be preserved from it. And you can see this really interesting statement here. Whoever had the courage to use it was forever free of the fear of contagion. And he showed this scar, right? So this was not accepted right away, right? It took a lot of convincing, but eventually it became the underpinnings of what in European, like Western medicine, would be used as, developed as vaccination. And like I said, very controversial. Cotton Mather even had a bomb thrown into his window, right? And there's this great note, Cotton Mather, you dog, damn you, I'll inoculate you with this, with a pox to you, right? So very controversial, even at the time. And I think maybe one of the most interesting players in this is Benjamin Franklin, who actually also had these fears, right? These fears about being vaccinated. And so he ended up losing a son because he had not had him inoculated. And he wrote this kind of touching thing in his autobiography that some parents might fear that their child is at risk by being inoculated, but he said that you can have that same regret either way. And so therefore it makes sense to choose the safer of those. And I think it's particularly interesting that the fear of vaccination, the fear of inoculation has been around just as long as the procedure, right? We have had to have courage to use it, even as our methods have become safer and safer and better. It still remains, many of these questions are just as relevant today as they ever were. Benjamin Franklin shows up another way. And sometimes people ask me what kind of science books I recommend for different age groups. If you have children who are kind of elementary school age and you want to give them one of the most exciting science books they could read, I would recommend this book, Mesmerized. It's amazing. It's about Benjamin Franklin solving this mystery, and you'll see how it comes into play with vaccination. So there was this guy, Mesmer, who was hypnotizing people, right? And he was having this dramatic effect on people in Paris. And when Ben Franklin was over there as an ambassador, he was asked by the king to evaluate whether or not this mesmerization was actually real. And so he invented a means by which he would blindfold the people who were being hypnotized, and was able to show essentially two things. One is that this was not, mesmerization was not real, right? But he also was able to demonstrate the placebo effect for the first time, that there was a way for people to feel as though they were receiving a treatment, even though there wasn't like a scientific basis for the, like a mechanistic basis for the outcome. So this was the first kind of origin of the double-blind study. And the double-blind study, of course, is a critical way for us to evaluate all kinds of treatments, including vaccines, which I'll talk about more in a minute. So check out that book. Then in the context of vaccines, I also want to talk a little bit about what they can do. And I want to talk about it particular in the case of polio, which is a really fascinating story. If you were to ask me what I recommend to your college age child, or to you maybe, I would recommend that you read this book, Atul Gawan's book, Better. It's an amazing book. I actually host a book club every time that I teach this engineering concepts applied to medicine class. And we read this book together, and we talk about this brilliant mind, this doctor, his observations on how to make medicine better in the practice. And one of the things he talks about is this incredible concept of the mop-up. Some of you will know that the mop-up, that's like how the World Health Organization describes this campaign to try to vaccinate as many people as they possibly can in a very short period of time when there's an outbreak of disease. And it's been a big deal in polio. So Gawan just goes out and actually does he kind of reports on the scene of a mop-up, and it's really quite amazing. Now, in my lab, we sometimes work with polio, so I'm going to show you just a snippet from the lab. Here we have some live cell microscopy. You're going to be able to see all of these different cells, which are just kind of a human cell line. And once they get infected with polio, they are going to start shining red. And you'll see it's quite intriguing, actually, as they kind of go along. Now you're going to see a couple of them are glowing red, and they're really going to start inducing the other cells, you see, to interact with them, to kind of even engulf them. It's quite amazing to watch what this virus can do as far as just controlling these host cells, right? Even with its limited genome. It's very fascinating to me, right? Frightening to most of us. But what it means generally is that if it attacks the nervous system, right, it can lead to paralysis, which is just a horrible outcome. So what happens in this mop-up, and the kind of latest outbreak, or the outbreak that Gawanda talks about, is in 2003 in India. They're going to try to vaccinate every single child that they can vaccinate. And just imagine what this is like. 50,000 square miles. They're going to have to try to vaccinate every single child that's under 5 years of age. And if it's less than 90%, right, they're trying to get this herd immunity, right? But they have a vaccine that's probably about 50 to 60% effective. And so their coverage needs to be extremely high. And so if you look here, Gawanda says, I asked him how many people that would involve. He looks at his sheet and he says, okay, we need 37,000 vehicles, all these vaccine carriers, and eventually we have to have them go door to door to vaccinate 4.2 million children in 3 days. And just imagine, it's a kind of an incredible feat. And one of the critical parts, just to go back to our second theme, which is kind of acceptance of the virus, is that it's very difficult. So sometimes people don't, are worried that the vaccine is actually going to give people the virus, right? Or that they're having more of they'll put their children more at risk by taking this vaccination. And there's this heartbreaking part at the end of this chapter in which a mother who has, I'll talk to you about that in a minute. First, let's talk about the two virus types. Or two vaccine types. So there are two different types of vaccine when you're talking about fighting polio. The first one was developed by Salk. And the idea there was that he inactivates the virus by growing it up and then treating it with formaldehyde, which damages its chromosome. It's genetic material. And so what happens as a result of that is that you have this virus, which is, I'll just call it dead for now. It's not going to replicate or anything, but it still can cause some kind of an immune response. Okay, and it's injected, and that was an incredibly effective vaccine for a long time. But then there was a second vaccine made by Sabin, and his idea was to continue to culture this virus in cells, like I showed you in my movie, but to do it at a low temperature, which induces mutation. And then to use that vaccine, try it out on, on monkeys, right? And as a result, to find out which ones would be protective without actually giving them polio. Now, what's really fascinating about a vaccine like this is it is live, which means that sometimes if you've been vaccinated with this particular vaccine, right, you can actually spread immunity, right? So it's kind of got an interesting, if somewhat rare benefit that way. It's also easier to, just the way that it's made, it's easier to be able to distribute. So when you're going, kind of backpacking with your supplies through India, right, you don't have to rely on an ultra cold or refrigeration. You can just distribute it. And in fact, you can even give it orally, which makes it much easier to administer. But one problem, in fact, is very, very rarely, but there is a chance of this attenuated virus becoming, a real virus, like an active virulent virus. And so very rarely there is a case where a person might contract polio from this virus. And that is the kind of thing that leads people sometimes to not want to take it, even though it's, we're talking about 1 in a million versus the risk of getting polio if there's been an outbreak. So there's, like I told you, there's this heartbreaking story where even though, you have this great evidence showing that bringing in the Salk vaccine and the Sabin vaccine has reduced polio almost to the point of wiping it off of the face of the earth. There was a mother who was just very worried about this effect. And by the end of the mop-up, her child had already contracted polio and wasn't able to walk. And it's this horrible part in the chapter where you just realize this is such a hard decision. But again, something that she shared with Benjamin Franklin, right, with countless others. So let's talk for a second about the other types of vaccines, just so I can get you into the mRNA vaccines, which is what you waited for. You've heard about these different polio vaccines. You're also familiar with the flu vaccine. Typically what's done there is, instead of looking at a whole virus, right, this is how we try to make these things safer. We go from looking at a whole virus to just looking at a part of a virus, right? So in this case, if you remember H1N1, the H stands for hemagglutinin, the N stands for neuraminidase. And so we can try to take those important parts of the virus and expose people to that instead of to the whole virus. Now, the advantage of that is you won't have as many side effects, but the disadvantage is that there's not going to be as high of an induction of immunity, right? So there's always these kinds of trade-offs to think about. And of course, the flu virus mutates so rapidly that it's really a very fast-moving game to try to, as you know, right, every year we have a different flu vaccine, and it's a very, very complicated process to try to guess what kind of influenza we'll have in the coming year. Then you have these live recombinant viruses, like the viruses against rabies. These ones do spread. We've modified the DNA in the virus. And so you can imagine that rabies vaccine can actually spread throughout an ecosystem, actually, once you've introduced it to a population. Excuse me. Then we come to some interesting things like these virus-like particles, or which I'll just go over quickly just to say, again, we can use something that is supposed to kind of make your body feel like it's experiencing this virus, and then to DNA. Okay, the DNA virus vaccines have been very interesting because you can try to just express certain parts of a virus, right? Like this, like they did in the case of SARS coronavirus. But the problem is that because it's DNA, it has the potential of interacting with our own DNA, which makes it a possible cancer risk. Now, these are, again, very rare, but these are still things that we're trying to think about. So what I want you to see through this is that we've been trying to work on technology for many years to try to make this safer and safer, right? So that people don't have to worry that they're putting their child at risk when they vaccinate them. And really, every year when I taught this class on viruses and vaccination, I would always say, and here is kind of our lodestar, if we could ever, by chance, make this technology finally work with mRNA's vaccines, then we'd have something really powerful. And amazingly, we are now in that era, right? So we have now gotten to the point where we can design RNA to produce part of the virus that would be recognized by the immune system. And in our case here with COVID, what we're talking about is the spike protein. Here's a representation of a coronavirus and some antibodies. And you can see these little tree-like looking things with the red poofy tops. Those are the spike proteins. They are essentially what is grabbing onto cells, inducing the internalization and causing a lot of our problems. And we're studying the spike protein rapidly. There was just a paper that came out lately where they were comparing the detailed protein structure of the spike protein in the original SARS-CoV-1 to like our current SARS-CoV-2 and finding those differences, find out what made it so that SARS-CoV-1 only caused something on the order of 1,000 deaths worldwide, whereas CoV-2 is just in a whole different level. So some of those studies are really starting to come out, but that's kind of what we're getting at, right? This spike protein is what we want to block, and so we introduced it into these our immune cells using lipid nanoparticles. I'll show you a little bit more about that in a moment. And then our own immune cells make the viral protein from that mRNA. And you can see a diagram of that here. Lipid nanoparticles with the mRNA are going in, they get incorporated, they get processed, they make this viral antigen. Okay, that's then carved up by the proteasome. And just like normal immune recognition, now what's going to happen is it's going to be presented at the cell surface. It's going to be recognized by our adaptive immune system. And that's going to trigger this large-scale immune response that helps protect us from being, from becoming sick. One thing several things are awesome about mRNA vaccines. One is that just designing the genetic material is so relatively simple. Just imagine, right? Moderna is doing this in 66 days. That's how long it took to go from sequence all the way to ready to go with the vaccine. Now, some of you are wondering, well, then what took longer than that? It's the testing, right? Figuring out if it's going to be safe and if it's going to work. But that is amazing. It's transformative. The second really great thing is that because it's RNA, not DNA, we don't have to worry about the virus itself, and we also don't have to worry about interacting with the host DNA. Okay, but one problem is that RNA is less stable than DNA, right? So that's why you hear about the shelf life and the ultra cooling with the RNA, because it's an unstable molecule. Okay, so does it actually work? Well, it's kind of amazing to see, these days, 1 year later, right, we have a pretty exhaustive picture of how it works. I want to show you here, if you can see where my cursor is, in this clinical trial of the Pfizer vaccine, there were 43,548 participants. Okay, so that is a massive amount. Part of the advantage, or like a silver lining of sorts, of how fast the virus is spread is just that we can have huge clinical trials. This is much bigger than usual. And they could be followed, right? And so you have a really good sense of how protective it is. And you'll see down here, early protection 12 days after the first dose and 7 days after the second, 95% efficacy was observed. So let's show that I want to show this graph in a little bit more detail here. So if you take a look, here you have people who received kind of this, this is Benjamin Franklin's double-blind study, okay? So some people received a placebo treatment, okay? And you can see here on the y-axis, the cumulative incidence, like how many people continued to get the disease. And here you can see the days after the first dose, okay? And so what you see is people who had the placebo, they just keep getting it and keep getting it and keep getting it, right? Whereas even after just a week or so, it's really tapering off who gets it, right? And by the time you've had the second dose, it is kind of astonishingly effective in preventing coronavirus. Are there any side effects? Yes, there are. So what you can see here, if you look on the left side, here are the side effects from the paper. If you look at the left side, you can see these different groups: dose 1, dose 2, how old, older on younger on the top, older on the bottom. And you can see the different side effects that occur, right? From soreness to fatigue, headache, chills, vomiting. They're all there, but I hope what you see is that, most people are, and so I should say here, green means very mild a side effect, blue means a moderate side effect. So almost all of the side effects are very mild or moderate, and they are, not that bad. So that's the amazing thing about this this vaccine. Now, some of you have probably heard stories about people who've had the vaccine and been really laid low. Yeah, for sure that can happen, right? You can see it. If you look down here, right, you can see more of these moderate cases show up. It definitely can happen. But the good news is you go through that for a couple of days and guess what? You're protected. And I think this is just an astonishing graph, right? When you see how this has particularly impacted aging populations from the New York Times just a few days ago, since we started vaccinating, the deaths among nursing home residents have gone from, just a huge, like if you take 100% being just this peak number here when we started, it's dropped down to something like a third of that already, and probably even more by this time. So while that cannot all be conclusively traced to the vaccines, I think it's a really good bet to say that a lot of it is. And so that's a very, very exciting development. Was the test population diverse? Again, we can go straight to the paper and we can see that. And if you take a look here, the answer is pretty clearly no. Right? It was not as diverse as it could be, right? I'm sure there was some effort made. I'm sure they were trying to do something, but there has been a call, and I think it's important to note, right? This is recent from the Washington Post. Really, we have to be thinking more about how we bring these things out in a way that really embraces diversity and realizes how important it is. You can see here, right here, scientists understood that speed and breadth of inoculation were equally important. They recommended a nuanced plan. Now, what the article goes on to describe is the fact that then people started worrying about these vaccines going bad. And so they adopted a plan of just get as many needles into as many arms as possible. And it sounds like a great idea when you think about just kind of knee-jerk reaction, but what it actually ends up doing is prioritizing people with more access. And so this is a thing that we're still actively having to think about. And I think it comes into play in a couple of other things we'll talk about in a moment. How long does immunity last is a question that a lot of people have had. And I think early on, there was kind of a misunderstanding that because antibody counts were going down with time, because those counts were going down, there was this idea that maybe that meant immunity was going down. Again, this is one of my favorite articles to recommend to people. Akiko Iwasaki and Ruslan Mezatov at Yale, they are I would say they're two of the people I most respect in terms of just incredible work and just meticulous. And they wrote an article for the New York Times many months ago, but it still holds, right? dropping antibody counts aren't a sign that our immune system is failing against the coronavirus or an omen that we can't develop a viable vaccine. And they have this to say, I recommend the whole article. I send it to friends and family members often when they have questions. But I would just look at this. You could say, this whole part is worth taking a look at. The antibodies produced in response to common seasonal coronavirus infection last for about a year, but the antibodies generated by a measles infection last for a lifetime. But what you have to think about is it's not just the antibodies themselves, right? So the amount of antibodies might peak and drop, but then you have these memory cells, right, that that are still around and ready to go and ready to build up another response. That's kind of the definition, right, of immunity for many diseases. And so that's a great reason to be very optimistic. Now, what this article doesn't include, because we didn't know it as much at the time, is the idea of mutation, which we'll talk about in a minute. But I want to answer a few questions that some of you have already submitted, and that will be one of them, so it's coming. So here's one of the ones that I got asked the most. Should people who are pregnant get vaccinated? When I first saw this question a couple weeks ago, I was pretty stumped, but amazingly enough, you're getting red hot off the press information today. But first, let me just start with, just like Benjamin Franklin said, there's a risk for not doing it and a risk for doing it, right? And you have to consider both of those risks when you're making a decision. And here's one thing that we know. Pregnant people are at an increased risk for severe illness from COVID-19 compared to non-pregnant people. That is a big deal. And also, they're at increased risk for other outcomes like preterm birth. So I'll show you some papers here that might be interesting. And what's amazing, they literally just came out like 2 weeks ago, right? So we've got great and solid information that's now come out. One is that SARS-CoV-2 has a higher infection rate. And look at this at the top here, 70% higher than similarly aged adults. And notice here, pregnant patients from nearly all racial and ethnic minority groups were overrepresented, right? So this was a diverse study, which is great because we get this very comprehensive idea. And here's what you see as their major conclusion. Pregnant women were not protected from COVID-19 in the early months of the pandemic, occurring in nearly all racial and ethnic minority groups. This data, coupled with a broader recognition that pregnancy is a risk factor for severe illness and maternal mortality, strongly suggests that pregnant people should be broadly prioritized for COVID-19 vaccine allocation in the US, similar to some states. Another paper that, look, February 23rd, right? So you're really getting this right in the moment, and this is straight from the original journals. Here they studied 4,000 pregnant women, essentially trying to see what the outcomes were of their pregnancy. And what they found, as you'll see here, is an increased risk of preterm and extremely preterm deliveries. And so the clinical implications of this work from those authors is that healthcare providers should recommend SARS-CoV-2 vaccination in pregnant women and women planning pregnancy, in addition to wearing a mask and those other things. Now, here's the problem. Much of that work happens from just a registry, right? Trying to follow people who are pregnant. What's really hard about this is that has been written up again just in the last week. There's what's called the pregnant people's paradox. They haven't been able to be part of these trials, right? For understandable reasons. They're worried about the safety. They haven't been able to be part of these trials, even though they have, a significantly higher risk of complications. And so this is something we'll have to think about when we start to plan out kind of our next phase of studies, et cetera. But again, in following people, Dr. Fauci recently announced that there are 20,000 pregnant women who have had the COVID vaccine without complications. And so that's quite remarkable. I think at least to my mind right now, the data is pretty compelling. Okay. Maybe the second highest question that people asked me was, what if you have allergies? What should you do? Should you still take it? We have a report on that. That is, if you look here, this is with the Pfizer vaccine in particular. And this is kind of interesting. You can see down here, I've been highlighting all the main texts in yellow for you. So in 1 week, they did a deep dive on this, right? Where it says here that about 2 million vaccinations were given, okay? And they detected 21 cases of anaphylaxis. Now, you could say 21, wow, that's amazing. I would say, yes, we need to be careful about it, but it's amazingly low, right? That's out of about, let's say about 2 million and about 20 cases. That means 0.001%, right? So it's extremely rare. In fact, maybe 3 zeros. I'm doing math on the fly here. So, but the key thing is it's extremely rare. And then it said here that most of these, if you were to read the article, most of these occurred in people who had a known, who would have anticipated it based on their medical history. And then also you see here, 71% of these occurred within 15 minutes of vaccination, which if for those of you who've already received a shot, probably they sat you down for 15 minutes just to make sure that you wouldn't have this kind of a reaction. So this seems like something that is again, it's always a risk. You have to consider these different risks, but it's a very low risk compared to the risk of contracting COVID, both for yourself and for the community. Another question people had was about human challenge trials. And for those of you who don't know about human challenge trials, the idea is an old one, right? The idea is that you would have healthy volunteers volunteer to be infected with COVID, after they receive a vaccine. The idea being that if you had these volunteers, that you might be able to make a lot of assessment very quickly about how safe it was or how useful it was. There's an article in Nature about it. I would recommend you to that because it's something fraught with different issues that are worth looking at, but it is complicated. In our case, we haven't really needed it because we've had so many patients. You saw 46,000, right, in that clinical trial. So I would say, and there are some problems. We did have this back in the old days, like Pasteur and his colleagues. They would, famously go to a country where malaria was present, and they would take these mosquitoes and they would put them in a jar and put it on their arm and let the mosquitoes bite the person who had malaria, and then they would put the mosquitoes on themselves so that they could see if they were able to get malaria in that way, and with different perturbations. And they would always say, we're doing this for the glory of France. It's kind of these amazing, and we kind of have this culture of that, all the way down to, Captain America, right? Trying this experimental thing. But it also raises some concerns, right? In a challenge trial, going back to one of our biggest problems, which is like the breadth, are we including underrepresented populations? Like we think about this thing with pregnancy, with older, by and large, these challenge trials have been done and are likely to be done with young, in this case, it's only happening in the UK. So young European men. And so it might not give us, right, the full picture that we're hoping for, among other issues. Okay, so now what about the new mutated viruses? It is an issue, right? So one of the problems with our overall global response is kind of the more people who get sick, the more chance this virus has to mutate, right? And in this way, I think sometimes people think, oh, if I don't get vaccinated, it's my choice. It doesn't really impact anybody. I actually think it's a little bit more like the idea of kind of smoking and secondhand smoke than people might think, right? Because it does impact. If the virus goes through you, the more people that it goes through, whether they manifest symptoms or not, the more chance it has to mutate. And the more chance it has to mutate, the more chance there is that a strain will arise that is more complicated, right? That is more complicated for us to treat. And so really we are depending on a certain large amount of the community to come together to be vaccinated, not only to keep everybody safe from, virus 1.0, but also looking towards the future and trying to make sure that we don't have a virus that's capable of shutting everything down again, right? So that we really can nip it in the bud. So let's talk about those mutations. There was, again, very recently, a great paper that you can find online, I recommend it, about staying ahead of the variants. And they talk about the different variants, which you can hear about in the news, but it's exactly what I just talked about. As more people become infected, the virus has more opportunities to generate mutations, right? And they have this kind of slightly poetic sense. Our dance with the virus will not be limited to these 3 variants. That's definitely true, and we already see it here in California with our new strain, right? So you can see too, preliminary lab data suggests that the Pfizer and Moderna vaccines are still effective against one of the variants. There's a worry that maybe it will be less effective against the others, but it's important to note that, and I guess I can show you some of this. So some people, this is a recent paper where people actually used a pseudovirus to try to test and see if the new variant could elicit antibodies from people who'd been, if it could be recognized, and what they see, what you should be looking here. Here's the reference strain and here's the new strain. In every case, it's pretty much the fact that there's like an equal response. So that was good news. And I think, for our new California coronavirus variants, which is being studied at UCSF, again, they're finding that the vaccines, even with a drop in antibody strength, they probably will be about as effective against the variant as they are against the original. So nothing to be afraid of yet, but we still don't want this to get outside of something that we can deal with. So it's super important that we think about our vaccination as something that we're doing as a population, as a world population, really, to prevent new variants, not only to protect ourselves today and people we know and don't know who the virus could be spread to, but also preventing new viruses from arising that could be equally damaging and harder to treat. Okay, so the recommendations based on all of that from Johns Hopkins, there were 4. Maintain policies that slow the transmission. This is what we're kind of trying to do. This is why different governments are organizing around this. Prioritize contact tracing and case investigation, something that we need to be doing, I would say, better as a group. Here's an interesting one. Develop a genomic surveillance strategy, right? So really start sequencing much more actively than we have been doing these different cases. And then start to coordinate that, going from genomic surveillance to trying to identify, okay, now I know the genome and I also know how virulent it is. Like I've characterized some of this virus. Okay, just a couple more, and then I'm more than happy to take more questions, but I want to make sure we cover some of the ones that you've already sent. How is the Johnson & Johnson vaccine different? Or, people have asked about some of the others, the Oxford vaccines or the Russia and China vaccines. I'll just cover a few here, right? So they're more traditional in the sense that the material that produces the spike protein is the DNA, not the mRNA, and it's introduced using what's called an adenovirus instead of these lipid nanoparticles. A disadvantage of the Johnson & Johnson is it's got this lower efficacy, right? 72%. It's still really strong, right? But the mRNA vaccines just happen to be higher. Remember, I told you that the polio vaccine was something like 50%, right? So when you have a lower efficacy, you kind of raise the coverage and you'll still get the herd immunity. And so it's still great. And for a lot of people, this is going to be preferred, especially because only one shot is required and you don't need to have the same storage requirements. The interplay between that advantage and disadvantage is a little complicated because, again, we have to be really careful. Like, let's say that we say, okay, we're going to take because this is only one shot and it doesn't require the storage, we're going to take it into the more rural areas, right? That's complicated because we don't want to have two tiers of vaccine access where one of them is less efficacious than the other, and the one kind of goes to people with access and the other goes to people who don't, for whatever divider. So we do have to really think about that. Luckily, people are thinking about that, but I hopefully will come up with a plan that incorporates those issues or that accounts for those issues. Again, just recommendations for further reading. Carl Zimmer, you might know, he's a great writer, a science writer for the New York Times, and he and Jonathan Corn put together a detailed article about how the Johnson & Johnson vaccine works. They actually have a lot of great resources there, so I recommend those articles. Here was one that I actually hadn't heard of before, so thank you to the questioners who gave me a chance to think about this. Are vaccinated people at higher risk for immune thrombocytopenia? Thrombocytopenia, it's a blood disorder characterized by low platelet levels, and because platelets can help our blood clot, it leads to easy bleeding and bruising. Normally, it's rare. There have been dozens of cases in vaccinated patients. So that could be seen as a cause for alarm unless you start thinking about it from a statistics point of view or what an expected value might be. Normally, from a group of 50 million people, which is how many people we've vaccinated in this country, we'd expect to find, because there's about 1 per 200,000 in the US, we'd expect to have 250 patients with this condition, even if there was no relationship. So that said, the arisal, like the fact that it arose just after, might be something to keep an eye on. And I think that I'm sure that's something we'll keep an eye on. But so far, very small numbers, and so far similar to expected normal numbers, this shouldn't be a reason to deter anyone from being vaccinated. Can vaccinated people still transmit the virus? Right now, looking again at the Johns Hopkins Resource Center, a similar question might be, do I still need to wear a mask, right? Or can I just walk around feeling like I'm a superhuman, immune to everything, right? No, we actually still do need to wear a mask and practice physical distancing for some time, right? Until we see the deaths dramatically increase. And I would say really just the jury is still out. I think there is some of the studies in Korea have been really interesting and telling, and I believe that there is a good chance that we might be able to find out that this would be safe to not have to do this once we get to a certain vaccinated level. But until we reach that level and until we know the facts, we're going to have to just be extra careful. And so that's the straightforward answer to that. And then the final question, so many questions kind of revolved around this one idea, right? Should I get vaccinated? Should I personally? I don't mean me. I mean, every person is asking me this question, should I get vaccinated? And, I'm not an MD, right? Everybody has a specific case, right? But I do want to say there is a lot of hope, right? So thanks to this incredibly, historically quickly developed vaccines, thanks to the fact that we can sequence the DNA so quickly, the genetic material of the virus, thanks to these distribution, just amazing feats of logistics, we are currently moving toward a place where we can at least look with some hope at the summer. And I wanted to bring this up, just looking at this. Widespread vaccination is the key, and not only widespread vaccination, but a continued reliance on these kind of masking and social distancing. We have a lot of reason to believe that, especially with today's announcement, about Merck joining the fight to produce vaccine, we have a lot of reason to believe that most of the country will be vaccinated by the summer, something which would really be transformative for this country. And hopefully, we can start then moving more towards helping other countries with this, because like I said, we all benefit by every single person that's vaccinated in the world benefits all of us. We're so tightly connected on this. And so I think there is a lot to hope for, but it does depend on us kind of deciding to go ahead and take that step and get vaccinated. So with that, I will stop my kind of formal remarks here. I'll turn the time over to Mike Friedman, who's the Director of Alumni Relations, and he'll moderate a Q&A, and I'm happy to take your questions.
Thank you, Markus, for that fantastic presentation. I know there's a lot of questions. We will try to get to as many of them as we can. I will call on you in the order that you raise your hand. Please, as a reminder, use the raise hand function in the reactions button. If you just raise your hand like this, I won't be able to see you. We ask that you be brief, and if possible, to turn your video on so that we can see you as you're speaking. We'll also select some questions that come in from the chat, and we did receive a number of questions, great questions during the registration, and I'd like to just ask, since you started with fear in some sense, let me go back to that question about fear. These vaccines were produced in an extraordinary short amount of time. What level of confidence do you have, and should we have, that we're not going to find out 6, 12, 18 months from now, or some further distance from now, that there are actually massive side effects that we should have taken into account in the first instance?
Yeah, I think that's a really interesting question. And the simplest answer to that, so of course, I can never rule out anything happening in the future, right? However, I do want to emphasize that mRNA vaccines, like I said, I used to lecture about this every year and just talk about the promise and the hope. It's not, even though it happens so fast for this particular virus, mRNA vaccines have been really in production for a long time, and their promise as far as reducing the amount of side effects is pretty clear. So I feel confident about that as a technology, while also recognizing that we continue to be surprised every day in science, right? And so I think we've definitely, though, made the best decision, and I don't think it's been a flash in the pan decision, right? This was technology that's been in the works for a long time.
Okay. The other question that comes up a lot, I know that a lot of people are concerned about schools and reopening of schools, and related to that, when kids are going to be able to be vaccinated. Do you have any thoughts about that based on the research that's been done or that will be done?
Yeah, right now, certainly there are children who are at higher risk, right? There definitely are higher children that are at higher risk, and we need to not consider children as just one group. But that said, the majority of children appear to have a different course of infection. I can give you an example from myself. I actually got coronavirus in March. I was teaching abroad in Paris, and we got it in Paris. And I had coronavirus for 7 or 8 weeks. I had that infection. It was very intense. My children also had it, but much less severe and probably over a few days, right? So it's just a very big difference in how able they would be to spread the virus, right? Because if your course is shorter, then your chance to infect others is shorter, right? Also just a much less threatening infection. And so there are many reasons which would tell you favorably that there are reasons we can open the schools. Now, that said, we have to make sure we do it safely, right? But and children will be vaccinated for sure, particularly beginning with ones with conditions. But I think that for right now, they clearly are in a different situation in terms of their immediate danger from the virus.
Thank you. I don't see any questions in the raise hand feature, so I'll go through the ones from the chat, and I'll pull also from questions that were asked previously. The J&J vaccine is DNA based. Does it have more potential cancer risk? And there are also concerns about people who are perhaps confused about the mRNA vaccines. and how that relates to DNA. And I'm wondering if you can talk about both of those things.
Yeah, those are great questions. So DNA, so first of all, just a quick overview. I consider the danger from both of these to be very, very low. When I am telling you the kind of possibilities, I'm telling you as a scientist is like, this is a possibility, but I think the risk, kind of like what we talked about in pregnant women, the risk of not being vaccinated is greater than the risk of the side effects. Okay, so just let me say that for both of these technologies. Like I don't foresee that Johnson & Johnson vaccine is going to be a large cancer risk. That said, so DNA is our genetic material, right? So we, everything like DNA is basically like a blueprint from the body telling you how to make us and how to maintain us. mRNA, and so it's in every cell, it's permanent, and importantly, if it's changed, particularly if it's changed in the wrong place, even if accidental, that can lead to something where a cell starts replicating and dividing out of control, which can lead to cancer. mRNA is kind of like, think about it as a molecule that took its orders from the DNA and then goes and does one of the jobs that you have to do. Like, so let's say that you have a gene that needs to turn on, right, and do its work, maybe to help you in some way. The mRNA takes the message, right, and converts that message into protein that can actually do the job. So it's a temporary molecule. It doesn't integrate into the DNA, and so for that reason, it's seen as a little bit safer. Well, safer in that with regard to that side effect, right? That anything, we're always worried to introduce DNA into a patient because of that chance, and in this case, a very small chance, but a chance of an interaction in the DNA level.
So there were a couple questions around masks, in particular around vaccination, wearing masks after vaccination. How long do you think someone realistically needs to wear the mask after vaccination? And maybe before you answer that, there was a question as well about whether masks work really at all, and if the virus can either escape through or come in through a simple piece of cloth.
There's no question that the masks work. That's highly established at this point in time. There are some amazing papers out there that have shown how the virus is spread, but clearly for a lot of systems, they've had to retrofit all of their air circulation systems, et cetera, just to try to filter better anything that we can do to try to catch this airborne virus. And better, in Boston, in New York, they've actually shown that these first responders, because they've been wearing the good masks, because they've been wearing protection, like you're at a lower risk in Boston, like at Mass General, if you work in the hospital with COVID patients than if you're just outside the general population in Boston. And the reason is that they're just taking much better care to protect themselves. And so it's very clear that masks work. And then you had a question related to that.
So how long after vaccination do we realistically have to continue to wear masks?
Yeah. I think the only honest thing for me to say here is that we don't know yet, right? I think the best practice for right now is to just continue wearing them when you're out in public. I have been, like I told you, I had COVID back in March. I've been wearing my mask and going out and distancing ever since. Some people have said, well, why are you doing that? You're already immune to it. And even though I do have a certain amount of immunity to it, I still think that it's really important that we as a community kind of commit to this, also to just make it easier for everybody to do that. And I think, so I think there are many social and health reasons to keep doing it. And I would say just keep going until we as a community, as a country, and as a world kind of decide, okay, looks like we've turned the corner on this.
Thank you. So we do have a question from Markham Cowrie. If you're here, we can unmute you, and you can have the mic to ask your question.
So I've seen a lot of questions, and people are asking the question around taking anti-inflammatories around the vaccine and whether that can mute the impact of the virus through muting the inflammatory effect. And that kind of correlates to a question: is a person's experience of the strength of their side effects, I call them primary effects because it's an indication your immune system is working. Do we know if there's any correlation between that and, the amount of immunity or the quality of the immune response of the individual person? And if you'll just kind of let me throw one more thing on the end of that, which is, these variants. My question to that is, how much can it really change before it becomes ineffective at actually infecting you anymore because it doesn't bind to the receptor or it doesn't properly interject itself into the cell and those kinds of things? I mean, isn't there sort of a limitation of those structures where they remain functional? They have to remain fairly similar to, the way they are now to continue to, be infectious in that manner. So, sorry to ask two questions, and answer one if you want.
And I hope you'll forgive me. I'll try to answer quickly so that we have time for the other questions. So the first question is about whether or not you can take like an anti-inflammatory while during the course of the vaccine and what that's going to do. I haven't really seen anything that suggests that you shouldn't be able to do that. You can, again, I'm not a doctor. I would definitely recommend anytime you do something like this and you have a specific concern, like maybe you're taking a specific medication, you should talk to your doctor about that, right? But I haven't seen anything. And you also notice, like in none of the vaccine protocols or whatever, are they telling you don't take ibuprofen, for example? So I think that's as far as I know about that. Then the second question was about mutation. What I would say to that, yeah, the virus is mutating all the time. So it's the combination of mutation and selection. So like you say, most of the time the mutation will either be totally inert, it won't make any difference, or it will be detrimental to the virus. So that virus will not be effective at reproducing. But sometimes it becomes more effective, right? And that's where we see these strains. The problem is we have given these strains a huge selection pressure, right? Because they have lots of exposure to people and they're passaging through lots of people. So there's lots of mutation, lots of opportunity, and then a selection pressure, which is just that there are humans around to infect. So that's the issue.
Thank you for being the first person to show your face, because now we have a whole slew of questions from people who want to do likewise. So we appreciate that. The next person is Kenita Watson.
Hi, I was wondering if the Johnson & Johnson vaccine might be more effective if given twice, like the Moderna and Pfizer are.
Yeah, that's a good question. I think for now, I would trust their guidance, right? So there are several issues kind of like that, Kenita. Well, another one that's similar to that is that since I've had coronavirus, should I be taking 2 doses or 1? And some scientists think that I should be taking 1, even if it's a 2-dose course, and others don't. These are kind of things that we're trying to figure out. But in the case of Johnson & Johnson, what we know is that the test that they did was with 1. And so we'll go with that. My guess is that they'll also be trying other things and making decisions based on that. And the kind of amazing thing is that we have so many people who we can try this on, that we should be able to get pretty decent answers on anything like that. But I would just trust the course they recommended, because that's the course that was specifically in the clinical trial. It's a great question.
The next question is from Maria Alcivar.
Hi, Maria.
Okay, hello. Greetings from Ecuador. So considering, I have a doubt. So when you get the shot of the vaccine, as in any other vaccine, you are expected to maybe have like mild symptoms, right? So in this case of this coronavirus, so when I get at the shot, how much time do I have to expect having those symptoms, or how does it work? That's my doubt.
Yeah,
Thank
no, it's a great question, and thanks for watching us all the way from Ecuador. That's great. So typically, what I'm hearing, so I showed some of the side effects, right? So some people just right away they have some redness or some soreness or fever. And then what you saw in the other side effects that I showed is that then some people have nausea, fever. Most common thing appears to be like just strong fatigue and a fever, right? That seems to be pretty common. And for some people, it's in the first dose. For some people, it's in the second dose. You know, you'll hear people say, oh, that second dose is a doozy. That's what I've been telling people is that's great. You should feel great because that is not going to kill you, for one thing. Unlike having the coronavirus, right? For me, I feel fortunate that I didn't have to go to the hospital, but there was one night where I could feel that my windpipe had constricted so much that I just thought, wow, I'm going to have to stay up all night and just see what happens, right? And that's not going to happen with the vaccine, right? You're not going to have to worry that it's going to cause real damage. You're just going to have to kind of experience these symptoms for at the most a few days, I would say. But what I'm mostly hearing, a lot of people are having nothing. This is anecdotal, but a lot of people are having nothing. Some people are having a pretty strong fatigue that starts 24 hours later and lasts for about 24 to 48 hours, something like that. Some people are feeling nauseous. What they're reporting is similar, but I would take that response any day over what I experienced, like losing my taste and smell, having these fever dreams, throwing up constantly, right? It's like a night and day difference just from my perspective as someone who had it.
Markus, there were a couple questions in the chat around mercury, around heavy metals in the vaccines. Can you talk about that at all? Mercury in vaccines has been a concern of many people for many years.
Yeah, that's interesting that you raised that. I haven't heard any problem with that in this particular case. That's basically all I can tell you. That's one you got me. That's one question I'm going to look up after this meeting. It's good.
The next question comes from Jake Smith. If we can unmute Jake, the floor is yours.
Hi, yeah, this is Jake. Thanks for taking the question. So we have a daughter-in-law who's pregnant, and my wife and I are going to go visit her. We've finished the 2-dose vaccine. We're about 5 or 6 days past the second dose. And we're curious, should we avoid seeing her on our trip to go see her?
Oh, that's an interesting question, right? In the end, it's so I think the key, if it weren't for the fact that your wife exactly was pregnant, I would feel confident saying go see your kids. We do know that, as I presented here, that pregnant people are more susceptible and more likely to be infected. So there's that complication there. Yeah, I can't make that decision for you. I could see where that's a tough one, but I think you balance the fact that you're extremely unlikely at this point to be, maybe the way to kind of to just be extra cautious with the masking, etc. I think that could be a good way to just try to balance your different concerns. But it's a good question, and it's a tough situation.
The next question comes from Ted Chang. If we can unmute Ted, the floor is yours.
Okay, yeah, thank you. My question relates to immunosuppressed patients and whether there's any data that an immunosuppressed patient who gets the vaccine, should they get antibody titers drawn to confirm that the vaccine was effective in developing a response?
Yeah, that's really interesting. I have not seen data on that particular population, and I think that But I do think that it makes sense, right? So if someone is immunosuppressed, obviously the problem might be that they're not able to mount a response, right? In the worst case scenario, you know, you think about somebody who's living with something like AIDS, for example, right? They might not be able to produce a response at all, right? And so, so I would definitely look at that. The one little caveat to that is that, you know, it's not just about the antibodies, right? So that's what I was telling you before. It's also related to T cells, et cetera. So just the overall immunity, I would probably say with any immunosuppressed patients, right? Just the rule of thumb almost always is to try to keep them away from anything infectious. And so that's, you know, that would remain my concern even when a vaccine was administered, with the caveat that I would just make sure, remember, I'm not a replacement for a doctor. I would definitely talk to a specialist about this if you're in that situation.
Okay, I think we'll take one last question from the audience, and then if we have time, I'll have one more for myself. Giovanni Anderson, if we can unmute Giovanni, then the floor is yours.
Hi, I have a question about the Moderna virus. My husband and I both got our first shots, but because of scarcity, they're thinking about pushing out our second shot. So how far should they push that? I mean, we were scheduled for a month, and now they're saying 6 weeks.
That should be fine. There, you know, if you remember, and I appreciate you bringing this up, Giovanni, because there's if you remember, like with kids, you know, typically we bring them in for a booster 3 months, 6 months later, right? And we're trying to get everybody that immunity faster, but there is some wiggle room in the delay. You wouldn't want to do earlier. Like I'd strongly recommend against saying, okay, like I got dose 1 yesterday, I'm getting dose 2 today, right? But the difference between a 4 or 6 weeks, that's acceptable. So I wouldn't worry about them pushing it out a little bit on that. Okay. It is true that what that means, unfortunately, is you have to be careful a little longer, right? So, so that part, I'm, you know, I feel your pain on that part, but I don't think that it means that you're going to have less immunity. Like that's not true.
Okay, last question. We started on fear. Let's conclude on hope and excitement. You mentioned that this is an extraordinary achievement to have these vaccines in weeks and then months when you include the testing. What, if anything, does that mean for the future of medicine? And in particular, what does it mean for people like you and your colleagues in the universities and also in industry about how you think about the kinds of advances, the kinds of medical advances that can be made going forward?
Yeah, I really appreciate your raising that question, Mike. So I guess I'll answer it with something kind of a personal story. I was asked to give a talk at MIT just before everything shut down. And I was explaining some of the work that we do and that we're trying to do. And one of the students raised their hand and said, well, that sounds like an unsolvable problem. And I said to the student, after thinking about it, something that I continue to think about, this exchange that we had, both the question and after when I talked to them, said, you know, that is an unsolvable problem, but we live in a golden age for unsolvable problems, right? And if you look, even just what's happened in our own Stanford engineering school or my Stanford bioengineering department, we have solved problems that I would have told you 10, 15 years ago were absolutely unsolvable. Super resolution microscopy, right? Optogenetics, some of these incredible problems that you never thought would be solvable by anyone. And the mRNA vaccine falls right into this scope, right? We live in a beautiful age for unsolvable problems, and I'm super proud of and thrilled by our incredible students who are basically being trained to solve unsolvable problems. And I think this should really give them some encouragement that what we're doing here is making a huge difference, right? Not just, yes, in medicine for sure, and in this particular case, but also if you just think about the whole world, about our civilization and what we're able to do and the challenges we're able to tackle with these students now, right? And with these incredible, the team that's here, right? I just saw it in the chat. It seems impossible until it's done. That's exactly right. So thanks for sending that. And it's the truth.
Well, thank you. It is 6:13, and so we have reached the end of our time, and then a little bit more. And thank you, everyone, for joining. Thank you for all the people who helped put this event together this evening. Thank you, Marcus, for shedding light on this complicated topic. It is, I think we all know, quite an anxious time, but also really, as you said, an exciting moment in the history of science and medicine. Thank you again to the audience for your time and questions. You will receive a recording of this event, as well as a very brief survey. So we ask you all to fill that out so that our events can be better and better. Thanks again for coming. Stay healthy, and good night.